So hello everyone and welcome as we return from our March in-person meeting back to our webinar series, and today it's on the topic of the role of cardiovascular proteomics. We're joined by two speakers, Professor Alicia Lundby and Professor Harald Mischak, alongside our guest chair for today, Professor Donald Jones. Donald will chair the question and answer session after both talks in a roundtable discussion with our speakers. So I just have a few of the usual housekeeping points to go over before we hand over to our guest chair to introduce the first speaker. So as always, we're using our Slack channel for questions and discussion. So please join us there, ask questions, use the thumbs up to let us know which questions you'd like to hear answered. And yeah, we'll be having the joint Q&A roundtable after both talks. So if you can direct your questions to each of the speakers by naming them, then that would be really helpful. For those of you who need an attendance certificate for this webinar, the details will be available on how to get this at the last slide. And so once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, the Young Proteomics Investigators Club, and the London Proteomics Discussion Group Committee for all their help and support in setting up this webinar. Thanks also go to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomics Research blog for promoting this event. And we're also really grateful for Imperial College London for providing us with the webinar support. Thanks again also to our YouTube channel subscribers. Harold's talk and the Q&A discussion will be available to watch again online after the event. And a huge thank you to our speakers and for our guest chair for their time today. So now Donald will introduce our speakers, but just a quick intro on Donald. So Professor Donald Jones is from the University of Leicester, and his research interests include identification of biomarkers for cardiovascular disease and the translation of candidate biomarkers to clinical use. So he's perfectly roundtable discussion with our speakers after both of the talks have been delivered today. So it's over to you, Don.
The second talk is going to be given by Professor Harald Mischak from the University of Glasgow, Professor in Proteomics and Systems Biology. He's also a founder and chief scientific officer of Mosaic Diagnostics, has in excess of 400 papers in this field and over 100 patents to date. And today he's going to talk about the urinary proteome peptidome depicting cardiovascular pathology. So Harald.
Thank you very much. I thought I somehow, one second please. I should present the slides. Why are you? Yes, here is the slide. Good. So thanks very much for inviting me. And I would like to talk about the urinary proteome peptidome in the context of cardiovascular pathology. I have an obvious conflict of interest. I own a few companies, which I founded myself, and I'm a bit less active now in academia. The talk is structured in a way that in the first half of the talk, roughly, I will present why I do things in a certain way, why we approach things, and in the second part of the talk, the application in the context of cardiovascular disease and the conclusions from it. And of course, the first question that comes to mind is why using urine? And I think you should keep in mind that urine, in fact, represents more or less the filtrate of blood. So urine is generated by filtration of blood in the kidney, and therefore we have all the information contained in blood, also in urine, sometimes in a slightly altered form, but ultimately it is available, and therefore there should be quite a lot of information about cardiovascular pathology as well. Now, the only thing that is missing is obviously technology to actually enable assessing the urinary proteome peptidome. The other issue I should maybe point out is that in a healthy kidney, you expect to see small proteins and peptides below about 15 kilodalton. You don't really see anything above 15 kilodalton. This changes in the pathological situation, but therefore in specific pathological situations. So in order to assess the information in urine as good as possible, we mostly focus on small molecular weight compounds, on peptides and small proteins. And after a couple of considerations, and I will show you in the next few slides what these are, the solution was really to use capillary electrophoresis, as shown here, coupled online to a mass spectrometer, typically a TOF instrument, and that enables separation and analysis of about up to 8,000 proteins and peptides within 1 hour in one sample. Since not everybody is familiar with CE, I would like to quickly explain or review how CE works. So basically what we have is we have a glass capillary in a diameter 50 or 75 micrometer that is typically filled for this purpose with a pH 2 solution. We inject the sample, 5% of the volume of the capillary, up to 10 maybe, and right in front of the sample we inject a high pH plug. And then on this 1 meter capillary we apply high voltage, about 25 to 30 kilovolts. What we see is that the OH minus ions migrate towards the beginning, towards the plus, and of course the H plus ions migrate in this direction. And we have stacking of all these peptides and proteins that carry both positive and negative charge. And in the end, we have stacked it very, very well. And this is also shown here. So in the beginning, we have, here you see the microamps, and we have higher or more microamps on the sample in the beginning to enable migration of the fast migrating ions. And this is roughly ended here, where separation starts. And then we separate based on really the charge, which is determined by the number of free amino groups, and by the mass, or let's say by the resistance to the migration in the liquid. It's fairly easy to combine CE with a mass spectrometer. Of course, we have to take care of the closure of the electrical circuit, but it's not really too difficult. In the end, it's similar to a nanospray interface. So here what we have is the CE capillary. This is on an orbit trap going in the sprayer and ending up roughly around here. We apply coaxial sheath flow here with about 200 nanoliters per minute, and we apply high voltage. Or here on the Bruker instrument that we routinely use, we have the same thing, the CE capillary here. Here comes the sheath flow. We have nebulizer gas if necessary, but we typically don't use it. And here, actually, the CE capillary or the interface is grounded, which makes life even easier. And this really enables quite stable interfacing. One advantage of CE in comparison to LC is really the reproducibility and stability. So here we have analyzed the same sample, a quality control sample that has actually been run routinely on a daily basis. This is basically a urine sample compiled of different urine samples from healthy volunteers. And I think we have about 40 liters of it, so it will last for many, many years. And as you can see here, here are different peptides listed, and the CV in intraday or an intraday assessment. And as you can see, the CV is quite low, which is important if we want to apply this in clinical testing. One of the reasons why we have such low CV is that we actually use a couple of peptides which we consider as housekeeping peptides, as interon calibrants. These are listed here. They are also published. So these are 29 collagen peptides that can be observed in almost every sample, and we can use all of these 29 to calibrate the data, both for the recalibration for the mass, but especially to calibrate migration time on one hand, and to calibrate for the abundance. So that this is similar to, let's say, urinary creatinine, just a bit more accurate in the end. When we now compare using the same sample, because this is a question I'm frequently asked, So here we have a urine sample separated in CEMS and here in LCMS. And you can see the migration or retention time here plotted versus the mass from 800 to 15,000 Dalton. Then you can see this is the separation in CE, and we have these lines. And these lines actually represent a different number of basic amino acids. Here we have no basic amino acid. Here we have 1, 2, and so on, because these are the ones that supply the charge in addition to the terminal amino group. And this, of course, is not observed in the LC. The separation here is a bit less ordered. If we compare proteins or in fact peptides detected in the same sample, then we can see a good overlap. But what we also see is that most of the peptides that are detected in LC, MS, are also detected in CE. MS, there's just quite a lot more to be detected here. I have to admit this was different, let's say 5-6 years ago. By that time, mass spectrometers were by far less sensitive, but now with newer, more sensitive instruments, we can see a really big advantage of CE. When we look at the mass distribution, we also at least get an idea where the difference comes from. It mostly comes from the low molecular weight range. And from a technical point of view, this is simply the result of not losing any peptides in the flow through. You may remember if you use an LC-MS run, then quite a lot of peptides, those that are small and are highly charged, will be in the flow through and are really not accessible for analysis. This is different in the CE, where we do not have any flow through. So we find all of these peptides, also the smaller ones, which gives a certain advantage. And another advantage is that the CE capillary is quite simple and can be flushed after each run with sodium hydroxide at high pH basically, and this cleans up the entire system. There is no carryover. While if we run an LC sample and then we do a blank run afterwards, then we always find quite a lot of signal in the blank run. And this, of course, generates issues if we want to perform clinical analysis, because we cannot allow carryover from the last sample to the next one. To sum up, there are certain advantages of LC, higher capacity. So CE can only be loaded with, let's say, up to 300 nanoliters. But this is not so relevant anymore, because by now I would say the analysis more runs into problems with respect to dynamic range of the instruments, given that modern mass spectrometers are quite sensitive. So therefore, loading more does not really help. Another issue is that we cannot run a shallow gradient on a CE. This can only be done on an LC. So if we want to go for increased runtime for better resolution in this respect, let's say run something for 6 hours, that's not possible. The CE run, the actual separation time is about 25 to 30 minutes. The advantage of the CE is the higher resolution, but much more even the higher reproducibility, that there is basically no difference between different batches of capillaries. We by now used this for more than 15 years, and we can still compare the data from 15 years ago to nowadays data. There is no flow through, and we can clean the system, eliminating carryover. Oops, sorry, that was too fast. Now I don't know how to get back. Okay, so on the previous slide, sorry about that, I showed what we generated in the next step, and it is basically a quite substantial database that now contains these CEMS data from different subjects, the clinical information, and this allows us to investigate association of different urinary peptides with different diseases. And this is what is shown here, the samples that are currently contained in this database. So we have, I think that's quite logical, a lot of samples and data sets from patients with kidney disease, by now a little bit over 15,000. But we also have 7,500 data sets from subjects with different cardiovascular diseases. And of course, plenty of controls and blinded samples from ongoing studies. Overall, a little bit more than 85,000 data sets. And this is the nice part about the CEMS or CMSMS, that all of these data sets are quite comparable, as long as, of course, they are urine and all sample prepared in the same way. And we can use all of these data sets to define biomarkers or to define peptides and small proteins associated with specific conditions, or also response to treatment. Trying to go to the next slide. Can I have the next slide, please? Thank you. So altogether, this high reproducibility enables to establish what I would call a proteome, or you can call it peptidome panel of urinary proteomic peptidomic data. So what you see here on the left side is actually individual analysis. Again, the migration time plotted versus the mass, and each of these peaks obviously represents a certain peptide or protein, but many of these are the same. And thanks to the calibrants, to these 29 calibrants, we can compile them and then also, of course, compare very large groups of data. And this is necessary since many of these potential biomarkers or peptides, in fact, show some but not a huge difference between different disease groups. One of the surprises really was that most of the peptides we found are, in fact, collagen peptides. So here I list the most abundant urine peptides. The abundance is presented as % of total abundance, based on a little bit more than 55,000 samples. And you also see the frequency, also the number of observations in these 55,000 samples. We have peptides from uromodulin, from fibrinogen alpha, but then most of the others are collagen, especially collagen alpha 1,1, also alpha 1,3, and there's one matrix Clar protein. And then later on, we also see additional fibrinogen, uromodulin, and so on fragments with lower frequency and also lower abundance. We do not yet know the reason why these collagen fragments are so abundant in urine, but I think one plausible explanation would be that all of these, when you look at it, carry hydroxylated proline indicated by the small p. And hydroxylated proline can not really be reused as an amino acid. It is most likely that hydroxylated proline serves as a signal to exclude these peptides from tubular reabsorption, and this is why they can be found in the urine. Now going from the technical part of the analysis to the search for biomarkers, as you probably know quite well, we have seen that single biomarkers are of limited specificity, and two quite famous single biomarkers are shown here, NGAL and C-reactive protein, which in the end, if upregulated, indicate something is wrong, but it cannot be really associated with a specific disease. The specificity or selectivity of these biomarkers is poor, and this is true for the others as well. In addition, they show quite high variability. But we can actually use a trick, and that is we can use multiple biomarkers. And this is, of course, supported by an analysis like CMS or actually any proteome analysis. This enables us to select multiple biomarkers. And in a study that was presented a while ago, actually aiming towards separating male and female subjects, which, of course, how shall I say, there is no clinical need for, but for an experimental approach, this does make a lot of sense because we have quite a high certainty on the assignment of sex, male or female. So there is less false assignment, very few false assignments in the group, and we can therefore be quite certain about male or female. What we see here is on the left-hand side, the number of selected features up to using all features in the sample, and from the back to the front, the number of observations, and here is the error in the independent test set, in the holdout set. As you can see, the more samples we use from back to front and the more features we use, the lower the error in the holdout set. To show this in maybe a more viewer-friendly way, here we show the same results based on 10, on 100, on 500, on 1500 features. As you can see, the error is reduced with higher sample size, but it doesn't really improve if we go beyond 100. If we use 10, if we use 100 biomarkers, we obtain even more accurate classification. And with 500, we actually have reached the maximum. An increase from 500 to 1500 doesn't really help anymore. So basically what we see is more biomarkers, more features, improved classification. And this is what we can do. We can combine multiple biomarkers into a classifier and then describe the disease in the best possible way. Not 100% accuracy, but at least fairly high accuracy. And this is what also was done in the context of cardiovascular disease. On one hand, in coronary artery disease. So what was investigated is whether we can separate patients with coronary artery disease from controls. We see the peptide profile up here, and down here those biomarkers that were selected. And in fact, they enabled separation with an AUC of about 0.87. Now, these were subjects with established disease. And ultimately, there is very little value in separating subjects with established disease from controls. What we really want to see is if we can identify disease at a very early point in time, earlier than possible with current measures, because basically the dogma exists, and I think this is correct, that early intervention would be the best approach in disease management. That also is true for cardiovascular disease. In the end, I think we can envision development of all of these chronic diseases in a way that we go from a healthy individual to ultimately death. We first have molecular changes and minor structural organ damage. And these minor damages, and certainly the molecular changes, cannot really be detected by clinical features, but they can be detected by using proteomics or peptidomics. Eventually, of course, organ damage will advance, and this is where traditional diagnostics comes into play, but then it's also too late to prevent organ failure and ultimately death. If we could detect here, we may actually change this trajectory and prevent the advanced organ damage. And in order to approach along these lines, we investigated whether there are proteomic or urinary peptidomic differences in patients that eventually developed an event, acute coronary event, versus controls that did not match controls. And in fact, there were a couple of potential biomarkers, I show them on the next slide, that could be identified, and if those were combined, then in fact, as you can see here, we could fairly well separate subjects that would have an event within the next 5 years from those that would not have an event. It is not perfect, but at least the hazard ratio of 8 is better than anything else that is available. And in fact, these biomarkers will or should now be tested in the intervention study, which I will outline a little bit later. Now I have the major advantage of not having to put an X to the biomarker, so I can actually disclose all of the information that we have. And what we see is not unexpected a deregulation in collagen fragments. We see a downregulation of especially collagen alpha 1,1 fragments associated with a future cardiovascular event. And we see an increase of collagen alpha 1,3 fragments and so some other peptides. Again, as you can see, the fractional increase or decrease of these is fairly small. It's highly significant, but nevertheless small. But by combining these into a classifier, we can separate these two groups quite well. We applied the same approach to heart failure, and of course this did make a lot of sense, because one of the key issues in heart failure certainly is fibrosis, so it was to be expected to find especially collagen fragments being deregulated as a result of fibrosis in the heart, and this should actually be different from fibrosis in the kidney. So what we did is we compared a little bit more than 700 subjects with heart failure to matched a little bit more than 700 controls. And then heart failure is separated in heart failure with reduced ejection fraction and preserved ejection fraction. So we also could separate in HFrEF, reduced ejection fraction, and HFpEF subjects. What was surprising is when you look at the slide now, so here are the most significant change urinary peptides, is we first of all see really a deregulation of collagen fragment. We do not see the downregulation of collagen alpha 1 that we saw also in coronary artery disease and in chronic kidney disease. We actually see an upregulation of these peptides here, but in some instances, like here, also a downregulation. So that's why I would call it a deregulation. The surprising finding was that this was the same in HFrEF and in HFpEF, and especially also in this group called HFmrEF, which is between HFpEF and HFrEF. So on this molecular level, on the urinary peptide level, we can obviously detect heart failure very well, but we cannot distinguish between HFrEF and HFpEF. And this is also true when we look in more detail. So here again are the top 20 most significant peptides and a graphical depiction of the deregulation, and you see this is all the same. And in fact, when we look at the log-fold change for the different peptides, we can see no difference between HFpEF, HFmrEF, and HFrEF. And also here we see quite a lot of overlap between the different peptides. We find more if we look at the complete, at the group HFrEF versus controls, but this is simply a result of having more subjects in this group, therefore more statistical power. Overall, and not surprising, this of course indicates changes in matrix metalloprotease activity in collagen degradation, but we also find links if we want to link to different biological functional pathways to the immune response and maybe also to IGF transport. Looking more closely in these data, and in fact, and then also asking the question, are any of these changes predictive of outcome, of unfavorable outcome? We could, in fact, in 326 patients, investigate this issue where outcome was known for more than 3 years. And we investigated especially the distribution of collagen 1A1 fragments in subjects with a favorable versus those with an unfavorable outcome, basically death. What we saw is that the C and N terminal collagen fragments that we observe significantly associated with disease and with outcome are upregulated, while all the fragments that come from the middle part of collagen 1A1 are downregulated, which was for quite some time a puzzling finding. But now I think we have generated, with the help of many others, at least a hypothesis, and that is that most likely collagen 1A1 is substantially cross-linked in subjects with heart failure, and the cross-linking has an impact on the degradation. So as a result of the cross-linking, we actually see or observe an upregulation of proteases, but they are only productive against the termini and not productive towards the central part of the COL1A1 molecule. Nevertheless, this should give an opportunity to maybe intervene earlier Right now, this is the window of opportunity for pharmaceutical intervention, but we feel, based on all of these studies we have, that we can actually detect disease at an early point in time, and as I said, then change this trajectory to something like this. And this is now being implemented. Hopefully, we're trying right now to get the financing for the study in a study called Preplot, where we take patients at risk. So these are type 2 diabetic patients with no evidence of organ damage. And then we randomize them 1-to-1. And in the early diagnostics group, we apply different classifiers that predict heart failure, coronary artery disease, or also kidney disease. And those that are positive will then be tested with refined classifiers to predict response to 3 classes of drugs, to spironolactone, SGLT2 inhibitor, and GLP-1 agonists, and then perform intervention and, of course, compare to the standard care. And I quite frankly think that this will result in a major improvement in outcome for the patients. Yes, I would like to quickly go to an attempt to explain the findings, which is hypothetical right now. Basically, we all assume that extracellular matrix and fibrosis is a key player in the context of certainly heart failure and also chronic kidney disease. And the assumption was for a long time that fibroblasts differentiating into myofibroblasts would then result in increased ECM production and increased collagen production, and therefore more extracellular matrix fibrosis. However, when we bear in mind now the data that we have, and when we look at collagen, in fact, what was observed a long time ago already is that advanced glycation end products are observed increased in collagen, especially increased in age and increased in certain diseases like diabetes. And at the same time, the cross-linking of collagen results in a huge decrease of the degradation process. So cross-linked collagen cannot be degraded or cannot be degraded efficiently anymore by matrix metalloproteases. So here the normal collagen, here the cross-linked. The same here for MMP3, I think, and for MMP9. So basically, if we now have increased cross-linking, if we have increased glycation in the collagen, then this will render the collagen fairly resistant to the physiological MMP9 degradation. And in the end, I think if we think about fibrosis, we really have to think about the two sides that play a role in the end, let's say, in the presence of collagen. On one hand, yes, of course, we have collagen production, but on the other hand, we also have collagen degradation. And if collagen degradation is impaired and production stays the same, then of course we will have collagen accumulation. So in the end, the balance between production and degradation is really what counts. And it seems that, in fact, in all of these chronic diseases, collagen degradation is impaired, is attenuated. And by interfering with collagen production, we will not really help the patient too much. So I'm quite convinced, based on all the data, that what we observe, the fibrosis, is not the result of increased collagen production, but it is a result of decreased or attenuated collagen degradation. Last but not least, I want to point out that all of these developments could in fact be turned into a proteome test that is now being used in Germany for now, and now very soon in more and more countries, I think. And I would like to conclude with a few statements that I hope I could present to you, since this is a proteomics talk, that CMS represents a very robust platform and especially well suited for peptide analysis in applications where we need a high degree of repeatability, like clinical testing. LC and C, I would say, are complementary due to the different separation principles. And what we observe is a variability in single biomarkers is not only true for CEMS, this is true for any biomarker. If you look at albuminuria or NT-proBNP values, you will see the same thing. But this can be counteracted by, for example, support vector machine-based classifiers that tolerate instability and inconsistency. Based on the data, I think it's safe to say that both heart failure and coronary artery disease are associated with significant change in urinary peptides very early, in fact, even prior to a clinical onset of the disease. And most of these are specific collagen degradation products, likely indicating fibrosis. Urinary peptides are, or many of these peptides are associated with fibrosis, and they may be of value as non-invasive biomarkers for fibrotic processes, and in fact, organ-specific fibrotic processes. And last but not least, I hope that early intervention guided by urinary peptides, as we're now trying to implement, may be key to prevention and management of cardiovascular disease. And I would like to end by listing the many people that contributed to this study, and thank you for your attention.
Thank you, Harald. Oh, sorry, Joe.
That's all right, Don. I was just going to remind our audience that, yeah, please use the Slack channel if you've got questions for our speakers, and use thumbs up with the questions that are already there if you'd definitely like those ones to be asked. And there will be the certificate of attendance at the end, but in the meantime, I will hand over to Donald now for leading the discussion.
Okay, thank you. And thank you, Harald, for an excellent talk, and again, another great set of data to discuss. There's a number of questions coming in, so if we split them into different areas, I guess, and the first one is about sample preparation. So for Alicia, did you perfuse the tissue before you proceeded with your sample preparation for the mouse samples? And how much of a problem were high abundance blood proteins in the samples from patients?
So blood proteins is a major cause of variation between samples, and it's something you have to pay really a lot of attention to. So we aim to get rid of as much of the blood as possible before freezing the samples. And just to tell a small story, actually, back when I was a postdoc, we did a project where we had 16 rats, where one person processed 8 rats, another person processed the other 8 rats, and we were sitting next to each other and preparing everything. And the end, when I analyzed the data, the major source of variation was the person who had been doing the perfusion of the hearts. So since then, which means essentially for the last 10 years, I think for me, it has been of primary importance to say, whoever handles the heart samples, it has to be the same person throughout. And we have actually done the same thing with the clinical samples, so that it's one surgeon, we know exactly where they have been collected from and how they have been handled after collection. Because I'm worried that if we do not do that, that otherwise the greatest source of variation will be the interperson variation and not the intersample variation. So get rid of as much blood as you can, and then I would say as long as the samples are handled as similar as possible, you know, then you can avoid the issue of it.
Okay, thank you. And Harald, a number of questions have come in for you regarding your sample preparation for urine. So I think, do you normalize? Could you give an overview of your urine preparation, and do you normalize for the volume?
Yes, I can try. When the preparation was actually optimized to be, how should I say, quick and simple, so that we have as low variability as possible. So what we do is actually we dilute the sample with urea and STS, basically to break open any protein-protein interaction and to have everything in solution. And then it's one ultrafiltration step that removes all larger proteins. That works quite well and easy. And afterwards, simply desalting on a PD10 column. to get rid again of most of the urea and the STS, and then lyophilization. And that has turned out to be highly reproducible and works very well, since as we look at the lower molecular weight range, peptides and smaller proteins, there is no issue at all. And now, what about normalization? Yeah, we use these internal standards to normalize the data.
Right.
And on the other hand, we have certain QC criteria, so we request, I think, right now about 1,500 peptides to be present, and at least 20 of these 29 internal standards must be present in order for the sample to pass QC. And that's it. That typically serves quite well. Sometimes, of course, urine samples are too dilute, especially if we receive inappropriate samples, mostly from diabetic patients where the collection was done in the afternoon and they're just too dilute. But then you can always use more or ask for another sample.
Okay, thank you. Alicia, when you looking at the data, you combine the omics data and the GWAS data. Was this an automated process which you gave rules to pull proteins of interest out, or are you manually integrating side-by-side data visualizations?
Yeah, no, it's certainly not an automated process. I would say it's almost a bit unfair just showing one figure summarizing the results, because every column was a lot of work to extract that information. But this was our way of trying to visualize huge amount of both analytical work and to summarize the finding in as simple a format as we could come up with. But it's not a tool. I mean, the GWAS itself is one analysis, right? And that was based on the UK Biobank. The single cell RNA-seq data was data that we acquired ourselves, and hence we have both data sets. Well, it's the single cell RNA-seq is all published, but that's analyzing all of the data, making sure that you can evaluate them in the same workflow. So we do Python-based data analysis. And then finally, the question about this, the FAIRS database are looking at intersecting with the drugs that are causing bradycardia and that target some of the proteins. That's also a whole study on its own. So we published a paper last year in JACC Cardio-Oncology where we established this linear regression model. And so it actually came out of, being under lockdown in Corona, not being able to do experimental work. We kind of, explored other directions. And I was just amazed to find out about all of the information that was actually publicly available in this FAIRS adverse events database. So it took, I would say again, a year for Robert and Johan, so an assistant professor and a master's student in the group, to set up this linear regression analysis. But once you have established it, then it's running another analysis, right? And then that is an approach to come up with which drugs are associated with a particular phenotype, and then for looking at which proteins they bind, it's again really evaluating data sets that are already published, right? So looking at drug-gene interaction screens and then setting a certain KD threshold for it.
Sure. And a kind of follow-up to that was, did you analyze was it a similar number of samples between the GWAS or the multi-omic data sets?
So I'm not sure I understand that, because the GWAS was on the 500,000 individuals in the UK Biobank, right? And the proteomics data was on the mice that was part of this study, which was 16 mice.
Yeah.
So yeah, I would say it's independent data sets where each independent data set has been QC'd and analyzed individually. And then so independent analyses and then merging the results of the individual analyses.
Okay, Yeah.
Mostly.
So the follow on to that was about, did you need to perform data reduction to resolve the PCA for the mouse data, I guess that is?
So for the PCA I showed was only for the proteomics data, right? So that we haven't done PCA across multiple data sets.
Right.
So we have not done data reduction to answer the question.
Okay. All right. Right. So on to data for you, Harald. So how did you manage the search space of your data for false discovery rate with respect to the absence of the specificity of an enzyme?
I'm not sure I fully understand the question. So it's more how I define the false discovery rate when I perform the sequencing.
Without, I guess, without tryptic digestion.
Yeah. I have to admit, I do not know exactly how this is technically done. Several of my postdocs did this. Sorry about it. One way to what we do as a kind of safety or so to assess the false discovery rate is we look at cysteine-containing peptides, because under the conditions we apply, we cannot identify any cysteine-containing peptides. So if you find cysteine peptide, then obviously this must be a false discovery. And based on the assumption that by chance every second peptide roughly should contain cysteine, based on the distribution of cysteine, you obviously don't want more than one peptide every 200 every 200 peptides of the false ones. And this is how we scan through the data. But basically, once you identify the peptide in the MS-MS sequencing, there's no need to identify it each time over and over and over again. So what we also did is we defined the data space of urinary peptides. And this is by now 21,600 and some peptides that are known and that can be found over and over again, and that are defined based on their mass and their migration time. Yeah. I think mass is 5 ppm deviation, and migration time is in a range of 1% deviation, and this basically gives a resolution of a little bit more than 10 million. So far, we did not find two different peptides at the same position. I mean, theoretically, they may exist, but so far, that's not an issue at all.
Okay, which is the great advantage of CE, yeah. I guess. Yeah, okay. Yes. And another question is about what post-translational modifications are you considering when searching your data? Are you looking for any unusual oxidations that might be interesting in the context of hypoxia?
Yeah, that's difficult because, yeah, as you pointed out, we don't have tryptic digest, so this results in the data space exploding. We typically allow for protein hydroxylation, but even that only on collagen and on no other peptides, and obviously methionine oxidation. But that's about it. We are now looking more into it. So we found a few phosphopeptides. So for example, fetorin is phosphorylated and refined phosphopeptides. And we find a couple of glycosylations as well. Right now, we only go for O glycosylation because that's at least something we can handle. We tried all kinds of other PTMs, but and we find them theoretically, but I would say all of these findings ultimately turned out to be incorrect, turned out to be artifacts. So this is a problem, but we have not found a solution yet.
Okay, no. Alicia, so some specifics about some of your findings here. It is known that some PKP2 mutations result in relocation of desmosomal proteins to the nuclei, which could potentially help explain some of your observations.
That's an interesting question, and we haven't investigated it, but it sounds interesting, and I would be curious to look into it. I think we have been considering that there are a couple of other genes that actually reside in the nuclear, by the nuclear membrane, where mutations can also cause a phenotype that is very similar to ARVC. for instance, lamin A and also TMEM43. And we would like to investigate if this convoluted shape of the nuclear membrane and DNA damage is really where the disease mechanisms merge. But as of yet, it's just speculation. So it'll take some years, I guess.
Yeah, I suspect so. Okay, so another question for you, Alicia, here. Did you see a response to your enzyme X activation in any other potentially fibrotic tissues, such as lungs or liver?
So this enzyme X activation, we have only evaluated that in the mouse model, right? So we haven't been able to give it to humans. And in the mouse model, PKP2 is specifically knocked out in cardiomyocytes, and it's done at adulthood. So the mice really presents with a cardiac phenotype, and we haven't looked at other tissues. So we have not evaluated it.
Okay. So another question for you, Alicia, about genetic variants. How about genetic variants in HCN4? They have been linked to SCD syndromes.
Yeah, I mean that is correct. I'm not quite sure what the question is on here, because I mean we looked at protein abundance and we have not evaluated yet sinus node samples from humans because they're very difficult to get hold of. So I have no data on correlations between genetic variants and protein abundance, if that is the direction of the question.
I think so. Yeah, I think so. You could potentially get that from GWAS data, I guess, couldn't you?
No, but only about the genetic variants, and that is certainly true. But I guess you then want to go to the protein level.
Sure.
To evaluate it.
Okay. I think there's a question about how you actually did the association between drugs and drug side effects and your protein hits in the bradycardia study.
Yeah, so I was alluding to it at the earlier question, in that it is really a study in itself to set up this linear regression model based on the data available in the FDA adverse events database. But what we essentially did was to analyze all of the adverse events in that database and find out for which compounds there's an overrepresentation of bradycardia events. And there were around 10 drugs that give increased odds ratio for reduced heart rate or bradycardia. So that's an analysis on its own. And once you have that information, you have to evaluate which proteins do each of these drugs bind. And it's not just, the reported primary target, but it's also off-target binders. And for that, we have gone to, again, other databases to evaluate the protein targets of the compounds, and then after that it was a lot of manual lookup in papers to evaluate the information that information was based on, to really make sure that it was only high confidence protein binding partners that we included in the end.
Right. Okay. Harald, there's a question here, and I kind of had a similar question myself about how do peptides from blood plasma compare to those from urine? Have you done that with blood or plasma?
Yes, so we investigated this. They compare, let's say, poorly. So overall, if you investigate association of blood plasma peptides with urine peptides, you hardly find a significant association. This changes when you limit it to collagen-derived peptides. So here we do find association. And this actually has then given also rise to the hypothesis, among many other observations, that likely collagen-derived peptides are excluded from tubular reabsorption. So you also see a huge increase in collagen-derived peptides when you compare blood plasma to urine. And as I said, you see a quite good correlation between these two. So I think the bottom line is that most of the plasma proteins, or peptides, I'm sorry, are in fact reabsorbed in the kidney. Of course, you could also envision that some of these are degraded, but this should also account for the collagens. But overall, I think the tubular reabsorption is really the explanation why we do not find a lot of plasma peptides in the end.
Okay, and there's a kind of follow-up to this. Endogenous peptidase activity can often leave peptides with ragged ends. So several versions of the same peptide differing in length by 1 amino acids. How do you deal with this challenge in your data handling?
Basically, I would say, thanks to statistics, I ignore it. Sorry about that.
Okay.
So if they have ragged ends and it's one peptide per patient, so to speak, then it will never become significant. And what we do is we typically request that any peptide be present in, well, 30, 50, or 70% of all the subjects in a specific group. Depends a little bit on the investigation, but if it falls below, then it will be just disregarded. And I think this is what happens with the ragged ends. So for example, at the same way, this may also be the reason why we typically do not find advanced glycation end products in urine, because again, they are here, but they are not consistent. And the variability then results in those peptides being rejected.
Okay, so as a general question to both of you, I guess, what proteomic MS technologies that are developed or not yet developed would help drive cardio research forward? So Alicia?
That's a difficult question.
It is a difficult question.
I think, my line of thinking is really centered on which types of samples are available, and I think Harald has done a very clever choice in focusing on urine proteomes. Personally, I'm interested in what changes occur in the muscular tissue, because it's different things that you can investigate, and from that perspective, I would say getting the freshly collected biopsies that I presented work on today is really limited in how many samples you can get. But at several hospitals, there are many more samples archived if you look at the FFPE samples that they have collected historically. So that's what we are focusing on now, is to really first set up a benchmarking QC method in saying, if we compare freshly collected with FFPE tissues, what can we see in one type of sample and what can we see in another type of sample? Because when we get a good understanding of that, we have the possibility of really studying many more samples. So I think I'm more focused on the samples, and then we have to develop the techniques that allows analysis of those.
Sure. Harald?
Yeah, I would fully agree. In fact, I would go even one step further and say the technologies are more or less here. It's really a matter of samples, of access to the samples, maybe a little bit of sample preparation protocol. And on the other hand, of funding. It seems it gets more and more difficult to get useful, good studies funded, because they obviously require a lot of samples. But it's a lot easier to fund all kinds of sometimes ridiculous investigative studies based on 5 samples with all kinds of claims. So I think there needs to be a rethinking here, and we really don't need to improve on the instruments, at least not for now. I think the instruments are fine. We need to remove the restrictions that are frequently associated with samples, as ownership on samples in biobanks where they disappear, and GDPR, which are 2 quite painful issues, and we simply need to run larger studies. to get decent statistics on it.
Well, I think that's a very good point at which to wrap this up. I think larger studies using current technologies with some of the understand some of the mechanisms that have been described today would be a very good way to proceed forward. So with that, am I handing back over to Jo?
Yeah, just briefly. So just want to say thank you to both our speakers and to John for chairing such a lively discussion. As mentioned earlier, we will have a form for those requiring certificates of attendance, which will be accessible for just a couple of minutes after we wrap up. And yeah, thanks again also to all our committee members who are working away in the background to make this webinar possible, and for everyone for attending. Just a heads up, our next meeting is an in-person meeting, will be hosted by Imperial College in South Kensington, London on July 21st. We have 2 keynote speakers lined up already with Oliver Shevchuk and Manuel Meyer giving talks, and please keep an eye on our website and social media for more details and more speakers nearer the time. And in the meantime, yeah, thanks again everyone, really brilliant session today, and we wish everyone a great weekend.